Good morning, all. Today we'll be discussing regarding necrosis and apoptosis. So, what is necrosis and apoptosis? They are basically mechanisms of cell death. क्या होता है जब होमोस्टासिस होता है, होमोस्टासिस होता है सेल बैलेंस इक्विलिब्रियम, जिस इक्विलिब्रियम में सेल रहता है. अगर कुछ चेंजेस होते हैं तो अडैप्टेशंस होते हैं. फर्दर इट इज इंजरी, अगर वो स्ट्रेस बना रहता है, अडैप्ट नहीं कर पाते हैं प्रॉपर्ली. रिवर्सिबल है सेल इंजरी, इसको रिवर्स हो जाए. अगर नहीं रिवर्स हो पाता है तो इट इज इरिवर्सिबल सेल इंजरी लीडिंग टू सेल डेथ. दो टाइप के होते हैं, एक्सीडेंटल सेल डेथ नेक्रोसिस, प्रोग्राम सेल डेथ दैट इज एपोप्टोसिस. Two principal types of cell death होते हैं, necrosis and apoptosis. Necrosis has historically been thought of as an accidental cell death, reflecting severe injury that irreparably damages so many cellular components that the cell simply falls apart. When cells die by necrosis, there is a local inflammatory response that clears the scene of the accident. In contrast, apoptosis is regulated cell death mediated by defined molecular pathways that are activated under specific circumstances and kill cells with surgical precision without inflammation or the associated collateral damage. The separation of necrosis and apoptosis is not always so clear. However, and some forms of necrosis are genetically controlled through a defined molecular pathway called as necroptosis. When we talk about necrosis and apoptosis, their features, we talk about the differences. Cell size. In necrosis, the cell will be swelling हो जाएगा, enlarged हो जाएगा. In apoptosis, there will be cell shrinkage or reduction in size. Nucleus, pycnosis, caryolysys, pycnosis, caryolysys. In apoptosis, it is fragmentation into nucleosome-sized fragments. Plasma membrane, it is disrupted in necrosis. In apoptosis, it is intact, altered structure, especially the orientation of lipids. Cellular contents, necrosis, enzymatic digestion takes place, may leak out of the cell. In apoptosis, it is intact, may be released in apoptotic bodies. No frequent adjacent inflammation is seen in necrosis, while in apoptosis, no adjacent inflammation is there. Necrosis is usually pathological. It is a culmination of irreversible cell injury, while apoptosis is often physiological, means of eliminating unwanted cells, may be pathological after some forms of cell injury, especially DNA damage. So we talk about differences in necrosis and apoptosis. Cell size will be enlarged in necrosis, reduced here. Nucleus, pycnosis, caryolysys in necrosis, while fragmentation and apoptosis. Plasma membrane disrupted, intact in apoptosis. Cellular contents, obviously, they are enzymatic digestion, with other contents will leak out. Here it will be intact, may be released in apoptotic bodies. Adjacent inflammation is frequent in necrosis, nothing in apoptosis. It is necrosis always pathological, while apoptosis may be both pathological and physiological. Necrosis, the main causes include ischemia, exposure to microbial toxins, burns, chemical and physical injury, and situations in which active proteases leak out of the cells and damage surrounding tissues, as in pancreatitis. All of those initiating triggers lead to irreparable damage of numerous cellular components. It is characterized by the denaturation of cellular proteins, leakage of cellular contents through damaged membranes, local inflammation, and enzymatic digestion of the lethally injured cell. Cellular contents leak through damaged plasma membrane into the extracellular space where they elicit a host reaction or inflammation. Some specific substances released from injured cells have been called damage-associated molecular patterns. These include ATP, released from damaged mitochondria, Uric acid, a breakdown component of the DNA, and numerous other molecules that are normally confined within the healthy cells and whose release is an indicator of severe cell injury. These molecules are recognized by receptors present in macrophages and other cell types and trigger phagocytosis of the debris as well as the production of cytokines that induce inflammation. Inflammatory cells produce more proteolytic enzymes, and the combination of phagocytosis and enzymatic digestion usually leads to clearance of the necrotic cells. Necrosis associated leakage of intracellular proteins through damaged plasma membranes and ultimately into the circulation is the basis of the tests that detect tissue-specific cellular injury. Cardiac muscle cells, for example, express cardiac-specific variants of the contractile protein troponin, while bile duct epithelium expresses a specific isoform of the enzyme alkaline phosphatase, and hepatocytes expresses transaminases. Necrosis of these cell types and associated loss of membrane integrity is reflected in increased serum levels of these proteins, which serve as the biomarkers that are used clinically to assess and quantify the tissue damage. Morphology. Necrotic cells show increased eosinophilia in HLE stains, attributable in part to the loss of cytoplasmic RNA and in part to the accumulation of denatured cytoplasmic proteins, which bind the red dye eosin. The necrotic cell may have a glassy, homogeneous appearance relative to normal cells, mainly as a result of the loss of glycogen particles. When enzymes have digested the cell organelles, the cytoplasm becomes vacuolated and appears moth-eaten. Dead cells may be replaced by large whorled phospholipid precipitates called as myelin figures, which are either phagocytosed by other cells or further degraded into fatty acids. Calcification of such fatty acid residues results in deposition of calcium-rich precipitates. By electron microscopy, necrotic cells are characterized by discontinuities in plasma and organelle membranes, marked dilation of the mitochondria with an appearance of large amorphous densities, intracytoplasmic myelin figures, amorphous debris, and aggregates of fluffy material representing denatured protein. Nuclear change appear in one of the three patterns, all of due to the breakdown of DNA. The basophilia of chromatin may fade, karyolysis, a change that presumably reflects loss of DNA because of enzymatic degradation by endonucleases. A second pattern, also seen in apoptotic cell death, is pycnosis, characterized by nuclear shrinkage and increased basophilia. Here, the chromatin condenses into a dense, shrunken basophilic mass. In the third pattern, known as karyorexis, the pycnotic nucleus undergoes fragmentation. With the passage of time, the nucleus in the necrotic cell totally disappears. We come to the patterns of tissue necrosis, coagulative necrosis, form of necrosis in which the architecture of dead tissue is preserved for a span of at least some days. The affected tissue has a firm architecture, has a firm texture. Presumably, the injury denatures not only structural proteins, but also enzymes. Presumably, the injury denatures not only structural proteins but also enzymes and so blocks the proteolysis of the dead cells. As a result, intense eosinophilic cells with indistinct Indistinct or reddish nuclei may persist for days or weeks. Now what happens? Ultimately, the necrotic cells are broken down by the action of lysosomal enzymes derived from infiltrating leukocytes, which also remove the debris of the dead cells by phagocytosis. Ischemia caused by the obstruction in the vessel may lead to coagulative necrosis of the supply tissue in all organs except the brain. A localized area of the coagulative necrosis is known as infarct. This is the localized area जो हमको infarct की तरह दिख रहा है, मतलब इसी को infarct बोलते हैं। और यहाँ पे हर जगह हमको eosinophilia बढ़ता हुआ दिख रहा है। Wherever जितना डेथ की तरफ जाता जाएगा, जितना डेथ हो जाएगा, वो सबका सब eosinophilia बढ़ जाएगा। Now we come to the second part, liquefactive necrosis. It is characterized by the digestion of the dead cells, resulting in transformation of the tissue into a viscous liquid. It is seen in focal bacterial or occasionally fungal infections because microbes stimulate the accumulation of leukocytes and liberation of enzymes from these cells. The necrotic material is frequently creamy yellow because of the presence of leukocytes and is called as pus. For unknown reasons, hypoxic death of cells within the CNS, central nervous system, often manifests as liquefactive necrosis. यहाँ पे localized collection of pus हो रहा है, abscess. This is liquefactive necrosis. Now, जब बात करते हैं gangrenous necrosis की, it is not a specific pattern of cell death, but the term is commonly used in clinical practice. It is usually applied to a limb, generally the lower leg, that has lost its blood supply and has undergone necrosis, typically coagulative necrosis, involving multiple tissue planes. When bacterial infection is superimposed, there is more liquefactive necrosis because of the actions of the degradative enzymes in the bacteria and the attractive leukocytes, giving rise to so-called wet gangrene. Contrasting features, very important question of dry and wet gangrene. Sites, जितना हमारे इंटरनल ऑर्गन्स है वहां पे ज्यादा वेट के चांसेस है, जितना लिम्स है एक्सटीरियर ऑर्गन्स है उस पे ज्यादा ड्राई के चांस है। तो साइट की बात करते हैं, ड्राई गैंग्रीन कॉमनली लिम्स पे होता है, वेट गैंग्रीन मोर कॉमन बावल। मैकेनिज्म, आर्टेरियल ऑक्लूजन, ड्राई गैंग्रीन, जबकि वेट गैंग्रीन में दोनों आर्टेरियल और वीनस ऑब्स्ट्रक्शन होएगा। मैक्रोस्कोपी में आर्टेरियल ऑक्लूजन है, उसको न्यूट्रिशन नहीं मिल पा रहा है तो ऑर्गन विल बी ड्राई, श्रंकन, ब्लैक, जबकि यहाँ पे वीनस ऑब्स्ट्रक्शन भी है तो दैट पार्ट विल बी मॉइस्ट, सॉफ्ट, swollen, rotten and dark. Putrefaction limited because arterial supply नहीं है, limited due to very little blood supply. Wet gangrene में it is marked due to stuffing of organ with the blood. Line of demarcation की बात करते हैं तो present at the junction between the healthy and the gangrenous part. In wet gangrene, no clear line of demarcation is there. Bacteria fail to survive. Numerous bacteria are present in the wet gangrene. Prognosis generally better due to little septicemia. It is poor due to profound toxemia. So just again we look, those are very important. Site क्या है? Dry का usually limbs रहता है, wet का usually bowel रहता है. The dry is only because of arterial occlusion. The wet is because of both arterial and venous obstructions. The organ is dry, shrunken and black in dry gangrene, जबकि soft, swollen, rotten and dark in wet gangrene. Putrefaction is limited due to very little blood supply in dry gangrene, marked due to stuffing of organ with blood. Line of demarcation present होता है junction पे between the healthy and the gangrenous part in dry gangrene. जबकि in wet gangrene, no clear line of demarcation is there. Bacteria fail to survive in dry gangrene. Numerous bacteria are present in wet gangrene. Prognosis generally better due to little septicemia in dry gangrene. Poor prognosis due to profound toxemia in wet gangrene. Now we talk about caseous necrosis. Caseous necrosis encountered most often in foci of tuberculous infection. The term caseous, cheese-like, is derived from the friable white appearance of the areas of necrosis. On microscopic examination, the necrotic area appears as a structureless collection of fragmented or lyse cells and amorphous granular debris enclosed within a distinctive inflammatory border. This appearance is characteristic of a focus of inflammation known as granuloma. ये पूरा border के साथ यहाँ पे, and if we touch this is cheesy fiber-like necrosis, caseous necrosis. Fat necrosis की बात करते हैं, refers to focal areas of fat destruction, typically resulting from release of activated pancreatic lipases into the substance of the pancreas and the peritoneal cavity. This occurs in the calamitous abdominal emergency known as acute pancreatitis, where pancreatic enzymes leak out of the damaged SNR cells and liquefy the membranes of fat cells in the peritoneum, releasing triglyceride esters that are split by the pancreatic lipases. Fatty acids are generated that combine with calcium to produce grossly visible chalky white areas, that is fat saponification. On histopathological examination, the necrotic areas contain the shadowy outlines of necrotic fat cells, basophilic calcium deposits, and an inflammatory reaction. इससे calcium वाला हमारा पूरा fat saponification दिख रहा है. Fibrinoid necrosis. This is a special form of vascular damage usually seen in immune reactions involving blood vessels. It typically occurs when complexes of antigens and antibodies are deposited in the wall of arteries. Deposits of these immune complexes together with plasma proteins that has leaked out of the vessels result in a bright pink and amorphous appearance in H&E stains called as fibrinoid or fibrin-like by the pathologist. You see, ये जो necrosis है within the vessel wall. This is plasma. ये blood vessel है. This is plasma. This is the fibrinoid necrosis. Now we talk about apoptosis. Apoptosis is a cell death that is induced by a tightly regulated suicide program in which cells destined to die activate intrinsic enzymes that degrade the cell's genomic DNA and nuclear and cytoplasmic proteins. Apoptotic cells break up into the plasma membrane bound fragments called apoptotic bodies, which contain portions of the cytoplasm and nucleus. While the plasma membrane remains intact, its surface components are altered so as to produce find me and eat me signals for phagocytes. As a result, the dead cell and its fragments are rapidly devoured before the contents leak out, and therefore apoptosis does not elicit an inflammatory reaction. Causes of apoptosis. Apoptosis occurs in two broad contexts: as a part of normal physiological processes, as a pathophysiological mechanism of cell loss in many different diseases. Apoptosis in physiological situations. Death by apoptosis is a normal phenomenon that serves to eliminate cells that are no longer needed, or as a mechanism to maintain a constant number of various cell population in tissues. It is estimated that humans turn over almost 1 million cells per second. It is very important in the following physiological situations: the removal of supernumerary cells in excess of the required number, supernumerary during development, involution of hormone-dependent tissues, on hormone withdrawal, cell turnover in proliferating cell populations, such as immature lymphocytes in the bone marrow and thymus, B lymphocytes in germinal centers that fail to express useful antigen receptors, and epithelial cells in intestinal crypts. Elimination of potentially harmful self-reactive lymphocytes to prevent immune reactions against own tissues. Death of the host cells that have served their useful purposes, such as neutrophils in an acute inflammatory response and lymphocytes at the end of an immune response. Apoptosis eliminates cells that are injured beyond repair without eliciting a host reaction, thus limiting collateral tissue damage. Death by apoptosis is responsible for the loss of cells in a variety of pathological states. DNA damage, radiation and cytotoxic anti-cancer drugs can damage DNA either directly or via production of free radicals. If repair mechanisms cannot correct the damage, the cell triggers intrinsic mechanisms that induce apoptosis. In these situations, apoptosis has a protective effect by preventing the survival of cells with DNA mutations that can lead to malignant transformation. Accumulation of misfolded proteins. Apoptosis can be induced during certain infections, particularly viral infections, as a result of the virus itself, as in adenovirus and HIV infections, or the host immune response, as in viral hepatitis. It may also contribute to pathological atrophy in parenchymal organs after duct obstruction, such as occurs in the pancreas, parotid gland, and the kidney. Morphological and biochemical changes in apoptosis. Cell shrinkage, cell size is reduced, cytoplasm is dense and eosinophilic. The organelles, although relatively normal, are more tightly packed. This contrasts with necrosis, in which an early feature is cell swelling and not cell shrinkage. Chromatin condensation. This is the most characteristic feature of apoptosis. The chromatin aggregates peripherally under the nuclear membrane and dense masses of various shapes and sizes. The nucleus itself may break up into two or more fragments. Formation of cytoplasmic blebs and apoptotic bodies. The apoptotic cell first shows extensive surface membrane blebbing, which is followed by fragmentation of the dead cells into membrane-bound apoptotic bodies composed of cytoplasm,